That's great.
Running into corals.
Nice.
There's corals everywhere. So
Nice
many.
following it.
That was fun.
That was very nice.
Yeah, massive
Really
I
colonies
enjoy that.
here. Yeah, this is spectacular. Yeah, back to the question earlier about climate change in the deep ocean. So it's certainly here, but it's not as well documented as what we hear about from climate change in a shallow reef. So the mass bleaching event that happened globally earlier in the year, things like that are not documented in the deep, and that doesn't mean that they don't happen, but we have much less ice here in the deep ocean. And the ultimate root causes are the same, so this is all from carbon dioxide going into the atmosphere and then going down into the water. In the shallow waters, a lot of the effects are due to warming, then causing bleaching of a lot of corals. In the deep, we have the issue of acidification and the schooling of the aragonite saturation horizon and deoxygenation, but much less well studied and documented because sampling and surveying in the deep ocean is so much less abundant than what we survey in shallow water habitats.
Yeah, but anything that's affecting the surface ocean is ultimately going to affect our deep ocean communities as well, because if there isn't as much productivity at the surface, that means less food for the animals down here at depth. And less food means less opportunity for growth of these really vibrant communities.
Yeah, absolutely. And the other big difference is in the deep, everything happens slower. A lot of these organisms are on the order of hundreds, if not thousands of years old. So if you do impact them, it takes a lot longer for them to recover. So they are not like weeds, they are more like your redwood forests.
What is that fish in the background?
It's
Oreo.
Oh.
How's that spelled?
Just like the cookie.
Okay, that's what I was curious
so
about.
it's a short form of the family name.
Okay.
It's an Oreostomatidae.
Does the eye kind of look like an Oreo too, a little bit?
I don't think that's what the name came from.
That's where I kind of got it from.
But yeah, they are really fun. And so we just call them Oreos for short, because no one wants multi-syllable names, especially since Oreo is such a fun thing to say.
So that was a large madrepora.
Yeah, it was.
That's
Yeah,
what
enormous.
we've got Steve on the line with these great IDs.
Yeah, and there was some, oh, there's a crab? See the red moving?
Oh, yeah.
Looks like a king crab.
Yeah, that looks like what Jeff was showing in his presentation.
Yeah, these are scavengers of the deep. We saw these come to the baited cameras. They usually show up later in the deployments because they got to walk there. So
I was going to say, are they slower?
yeah,
Go for a zoom there.
they're determined, but they're a little bit slower. So this is Lithodes longispina. It's got really long spines, hence the longispina.
Is that a globally distributed species? I thought I
Think so.
saw that in the New England seamounts at some point.
Spikes.
Yeah, there's
It's like it would hurt.
right. Yeah, it's like don't touch me. That's really cute, though.
All right, come on.
Steve says there's a benthopectinidid
Come on.
star.
Come on, Aaron. Yeah, sorry.
Yeah,
I
that's
didn't
the
hear
name
you.
of that star that I
Ah,
couldn't
there we
remember the
go.
name.
Nice.
I was like, it starts with Ben something.
Nice. And a small stylasterid between them.
Santiago says that this is such a cool dive.
So much. I don't know what to look at next.
I know.
You know?
It's really challenging. You want to like look at something new and exciting, but there's so much to look at. Now imagine if you have to draw a box around every single animal you see. Because that's what we'll be doing. So while we've been annotating, we've been doing localizations of these animals, meaning that we draw boxes around each animal in order to feed this data into FathomNet, a repository of animal identifications that'll help machine learning to be able to assist us with annotating deep sea animals.
Yeah, and for context, what Megan is referring there to, so yeah, that process on terrestrial habitats is already quite well established, and there's a lot of these machine learning that are used to identify a whole bunch of things. But that process requires having good reference libraries where a human verifies what it is. In the deep ocean, we have very, very
Another fish.
scarce reference libraries.
Another complicated factor with that is lighting, I think. It's really hard because vehicle systems are flying at different depths and lighting configuration is different, so it can be harder to identify.
Here.
Yeah, and the quality of the imagery can be highly variable.
Yeah.
This is a Diplocanthopoma. It's a related to the Cuskills. It's in that same Ophidiiformes, but it's a bifidid, the family Bifidinae.
Nice.
Yeah, and if that wasn't enough with lighting,
Do you want me to
the
go away?
plasticity of some organisms makes it very even harder. So some sponges and a lot of corals too can look very different depending on what environment they're in. So you can have the same species looking very, very different. And conversely, you can have different species looking very similar in convergent evolution. So a lot of difficulties and impediments to kind of automating that workflow.
And it might not always be obvious what the like important characters are for identifying a species. Sometimes it's branching pattern, or sometimes it's color, or sometimes it doesn't have scales on its snout in a very particular pattern. There are a lot of factors to consider, and this is just like the knowledge that we've gained over many years of understanding it, and how do you explain that to a computer that understands data in a very different way than the human brain.
It's a Chalogorgia fan. Many crabs.
I love the squat lobsters with their giant arms. They're great. They're the sternostylus.
Yeah, I think this was a pretty good dive for a first dive of this expedition.
I would say so.
I think we kicked it off with a nice bang.
Definitely.
We're going to have to outdo ourselves somehow.
Yeah, and we got a lot more action to go, so another hour here on the bottom, and then we'll be moving about 30 miles northeast of us, another seamount. That one had been mapped before, and so we'll be planning to dive there later in the day. And then conducting kind of the same exercise here over the next 3 weeks. About 12-hour dives or so, and then moving on to our next spot. So we've picked out a little over a dozen spots all around the Palau National Marine Sanctuary, covering different habitats and different depth ranges. And you all can join us on these fascinating explorations.
How many viewers do we have online at the moment? Is that something we can still see?
I'm not sure how to check that. Sorry.
Oh,
You'll
it's
hit
okay.
view live visitor stats. So just click on that.
Okay.
Sometimes it's cool to see people tuning in all over the world since we're doing 24-hour operations. It's pretty early in the morning in Palau, but it might be afternoon or evening or daytime elsewhere.
I want you to have a podcast. You have the nicest voice.
Yeah, I was just thinking the same thing.
I would listen to that all the time. Yeah, so there, right now, so right, that's the one where your mouse is, the 121 visitors online right now. And then you can also see over to the right, you can see what countries people are from. So there's quite a few different countries tuning in right now.
Nice.
You also have different languages, and it provides, yeah, quite a bit of information. Looks like most people are tuning into channel 3. It's got 4 different views.
Wow, this is really cool.
We're going to continue
We're going
east.
060.
060.
Yeah.
Yeah, thanks, Vidya, for walking us through that.
Yep, no problem.
Well, something in the Atalanta camera.
I think it's a siphonophore. Now we get to see the backside of that madrepora.
Yeah, it's stunning.
Looks like there's some gooseneck barnacles on it when we looked at it before.
I was going to ask you if that's what that was.
Yeah.
I didn't realize. So they attached to coral then?
Yep.
That's pretty cool.
Mm-hmm.
Do they attach to other different organisms?
Oh, I've seen them on sponges and things too, yeah.
Oh, cool.
And sometimes on crabs.
Not the same ones that carry around the coral.
No, generally not. But yeah, some of the bigger crabs will have gooseneck barnacles on them. I've seen it on some urchins in shallower water too.
What other deep sea creatures carry things on their backs? Like we've seen the anemone backpack and the coral.
Scuba divers.
Yeah, true. I mean the deeper inhabitants, I guess.
Well, you know about the Pharonoma, the inspiration for Alien. It carries around a salp with it. It's a type of amphipod.
Huh. Okay.
Yeah, and it just like kind of eats out the inside and lives inside of it.
So less of a decorative, stylish touch. Less the symbiotic, more parasitic.
More parasitic.
Yeah.
The deep staria.
I know, this one is one of my favorites.
Yeah, it always has that parasitic red amphipod in it.
Is it parasitic?
It is parasitic. It's also
that was
blind.
determined? Because when we first got this really incredible footage of the deep staria in the Revillagigedo archipelago,
that was my internship year.
Yeah.
I was on watch during that, and
we were together, weren't we?
Completely blew my mind. Yeah, it was really cool. But the experts all at the time did not know the relationship between that crustacean that was living inside of this giant medusa.
Yeah, there was a paper written about it not too long ago.
Oh, cool.
Ooh, let's look at this sponge. That's
Nice.
weird. It's a little branchy white sponge.
Which one?
The white branchy thing that
Here?
looks like a coral, but it's totally a sponge. Yeah.
Go for a zoom.
Steve was asking exactly the same thing. What is that white coral underneath?
It's not a coral. It's a sponge.
Yep.
Isn't that weird? And the sponge has, looks like little anemones growing in its tissue as well. And there's a ton of amphipods on it. Some brittle stars.
Right.
Oh,
And an isopod. And a shrimp. And some hydroids. A squat lobster.
that's a lot. Isn't that? You see how fast you can say everything.
Right?
Goes up at the end of the tether.
Yeah, I will get us moving.
Do you want me to pull wide?
Bridge
Yeah, you can
now.
come on.
Yep.
Can we move 20 meters 060? Thank you.
Speaking of internships, that application's open right now.
Yeah, it is.
I would highly recommend applying. That's how I started on the Nautilus, and I think that's how a lot of
Me
other
too.
people did as well.
Definitely.
Do we know when the internships close?
Yeah, January 3rd.
Okay.
So yeah, thanks, Aaron, for pointing that out. Highly encourage folks to apply for both our science and engineering internships and our science communication fellowships. Both of those applications are, the application packets are available online at nautiluslive.org/education. The internship applications close January 3rd, and the lucky candidates will have a chance to sail on a Nautilus expedition next year. And we got a very exciting season next year that will start in Guam and the Mariana Islands, then go on to the Marshall Islands, then proceed to the Solomon Islands, and then end in Fiji, so going across several archipelagos in the Western Pacific.
It's going to be a good year.
It's going to be, yeah, really good year.
I'm really excited.
It's a really hands-on internship, really supportive.
before I was an intern, I was watching Nautilus live, so you never know. This could be you.
A viewer is asking if we've seen a bubblegum shark.
Oh, a bubblegum shark? Huh.
I've never heard of that common name.
Never heard of it.
There's
either.
bubblegum corals. We've seen lots of those, Paragorgias. But that would be cool. Maybe it's something we don't, maybe it's something shallow in this region. I don't know. I'm gonna Google it.
There are a lot of sharks around here. We haven't seen one yet today on our dive, but during the baited camera deployments, there were a number of different species of sharks that showed up. So I do have high hopes for this expedition of seeing quite a few different types of sharks. A lot of times the sharks do get interested in the ROV, and we often see some at the surface when we're recovering the ROV. We'll often have the oceanic whitetips will hang out with us on our recovery. They're quite curious and like to chill.
Yeah, just to add a little bit of context there, the baited cameras that Megan is referring to, so back in February and March, there was an expedition by National Geographic Pristine Seas. And yeah, very thankful to our partners there that have shared the data and shared and helped us with planning this expedition.
Go for zoom.
Oh, wow.
Yeah, look at these benthic ptenophores.
Yes.
Wow.
Did they get stuck, or are they there on purpose?
Oh, they chose to do that on purpose. So they're specifically modified to live on substrates, and we see them growing on corals and sponges. That gets them up off the bottom so that they can use those long feeding tentacles.
Oh, Steve would like us to collect some ptenophores. How are we doing on possibility for that?
Yeah, we are not moving, so we could do that.
What do you think, Daniel?
Yeah, so if we're set up to do so, I think that's a great idea.
Come on. So
Does
And
Steve want us to?
With the Chrysogorgia, yes.
Yeah.
So,
For preservation, do we want to fix some of them in formalin? Just throwing that out there.
Yeah. Normally we just use ethanol, but let's see what he says.
I know
This is
sometimes
a
these
co-request from Steve Oskovich, scientist ashore, and Alan. So I guess would a snip with one of these ptenophores be the best strategy here?
I think so.
Yeah, I think they might. Yeah, maybe a branch that has a couple. So yeah, Steve mentions one in formalin and one in alcohol.
Okay.
I think this coral jake has a couple branches, and I think that the part that we're looking at closest to us would be the best spot.
Okay. Trying to get stable here.
And Chrysogorgias tend to be a little negative, but they can be a little floaty.
All right.
The sample
But
cameras?
they're very flexible.
So snip and slurp, right? Or
I don't think it'll go through the slurp.
okay.
It might get stuck.
They're a bit hard.
This one's very bushy.
Okay. All right.
Then if we snip, then let's go for the starboard bio box. Maybe F, like the most aft large bin.
Can you push in a little bit, Aaron? That's good there.
I think you're going to want to get more than that. So like actual branching points of,
Okay.
of the Chrysogorgia. So I think come from the top and grab around the branch that's close to us. Yeah, like that.
Okay. Here?
Does that
down?
look good, or do we want less?
Less?
I think a little bit less might be fine.
Okay.
Yeah. We really need kind of like one, basically two, like one in formalin and one in ethanol. That should be
I
good.
think that looks good, yeah.
Yeah,
Okay.
that's probably plenty. Hopefully they don't fall off. Nice.
No,
Okay.
they'll stay attached.
All right, so, yep. Starboard, aft, box.
Okay, swinging around. If you can hit the sample salvo. Yeah. And then can you also hit the craft button on the bubble?
All right.
PTZ. There we go. All right. You can bump out the box.
All right. Box coming out.
Okay, so
Thruster
the
wash.
aft one, right?
You might want to secure the thruster.
Okay. Let me just...
It's just a little bit sticky, isn't it?
Yeah.
Close it up.
Close it up, yep.
Great job, Jake.
Yes.
So how do we preserve the samples?
Yeah.
Yeah, so each sample has a different preservation method, but typically with the biology that we'll be working with here, we'll be preserving it in ethanol, which is basically like alcohol. For this one, we'll preserve one part in formalin and one in ethanol. Sometimes we'll dry samples depending on the type of coral. Rocks, of course, are just dried. And for eDNA, the water samples that get filtered, the result of all that filtered water is just a filter, and that filter will get frozen, and then later the analyses that will be done with that, all of that material left on the filter will have been preserved by being frozen. So we have a few different chemicals, freezers, fridges, things like that to preserve.
Thank you.
No problem.
Yeah, and on the reason for formalin ethanol, so ethanol is broadly used. It preserves DNA, which is obviously one of the more newer methodologies for analyzing and classifying. However, it does take the color out, so things will start losing their natural color, and they also will become hardened. So you preserve the DNA, but you lose the shape and color, and that's why the reason of preserving in both the ethanol and formalin. When you preserve in formalin, you don't lose the color as much and the shape. So when you have multiple preservatives, you can have different analyses that you can still do afterwards.
Okay, there's a question from a viewer. So how do deep sea plants reproduce?
Go for a zoom there.
There are no deep sea plants. Oh, it's an Iliophis.
Iliophis.
Oh.
Ooh, head shake. Wiggle.
They always shake their heads.
They,
In disagreement with the ROV.
Yeah. They're like, oh my goodness. This is an arrow-toothed eel. In the family Synephobranchidae.
Cutthroat eels.
All right, come wider.
Yeah, just a little context to the question there about the plants. So, yeah, plants rely on sunlight to photosynthesize, and for all effective purposes, the sunlight is gone after about 200 meters or so, and then deeper than 800 meters, you have absolutely no light that is detectable by human eyes. So yeah, the algae, the marine plants, the deepest are several hundred meters, and then after that, you don't have any more plants.
but maybe the viewer was wondering about all these corals, which look a bit plant-like.
Yeah, thanks, May, for that. And different strategies. So different corals do different things. So there are both. On one hand, you have either gonochoric versus hermaphroditic. So hermaphroditic, where you have a single colony producing both eggs and sperm, and a gonochoric, meaning when a single coral is either male or female. So you have those two things, and then you have either spawners or brooders. So and spawners are those corals that basically release their eggs and sperm into the water column, and then fertilization occurs externally. And brooders will retain the eggs, and then fertilization will occur internally. And there are some trends. So for most shallow water hexacorals, they are hermaphroditic spawners. You have these mass spawning events in shallow water coral reefs where you can see the spawn aggregating at the surface. For most deep water, particularly octocorals, they appear to be gonochoric brooders, but very little studies. I mean, this is something that's virtually unknown. You know, as we alluded to earlier, most species are yet unknown, and besides not knowing, we know very little about the life history of these things, how they reproduce.
Like 5 meters on the delta.
But it is a
Down, yeah.
important question for managing these organisms.
And coming back to the how often do we find new species question, Steve Oskovich, one of our scientists ashore, who's been giving us lots of great IDs, has shared that collection we just made of the Chrysogorgia with those I'm not sure if I'm going to say this right, Platyctenids, which are those sort of pink gelatinous associates that we were seeing, those ptenophores, might be a new genus or species, and that they're not really common on Chrysogorgia, so could be really finding something new here.
Yeah, I've never seen them purple before. They're usually like pink or orange.
He says even closely similar species morphologically can have large genetic differences, so I think that's pretty exciting. We've got really nice video, and now we've got some good samples.
That's really exciting.
I like that Anthomastus tahinotis down there. That's the really long body that.
Yeah. There's lots of good stuff.
I've been seeing those ptenophores on like almost all of these bottle brush Chrysogorgias that we've been seeing.
Yeah, I can see it in the cinema cam.
Oh yeah, the cinnamon cam's good at picking up those long feeding tentacles too.
Wispy.
So just quick time check. So it looks like we would have to leave right around 3:30 local times. Is that right? So in about 35 minutes.
Okay.
For to be on deck by
4 o'clock.
By 4 o'clock, yeah.
Sounds about right.
a question from one of the viewers. Do you have a website with references to scientific papers based partially or completely on your expeditions, and so they could see the results?
Yeah, great question. So on our Ocean Exploration Trust website, nautiluslife.org, we're starting to build out publications that have been published over the years, so 15 years of Nautilus expeditions. We have close to 350 peer-reviewed scientific papers that have been published from our expeditions, and yeah, and it's a growing number. We have not yet made most of that that part of the website public, but we're starting to build that out. We also publish every year an annual report, and that has all the peer-reviewed publications that were published that year. So there's a couple different ways, but yeah, our website already has or will have that information soon.
All right. Now here's a Halloween question. So, if you were a fish, what creature would you be most terrified to encounter and why? So I guess I could start with that question. If I were a fish, I think I'd be most terrified to see or encounter a shark, only because I do not want to be eaten and I would still like to swim away.
If I were a fish, I'd be most terrified to encounter a fishing line, because I'd probably be dumb enough and go for it.
I feel like mine would be one of those angler fish, because I'd like be like, oh, look at the line.
It's so beautiful.
It's so beautiful.
Quite
think-
the hairdo.
I think I'm on the same page as you. That one's got quite a few of those as well, doesn't it?
Mm-hmm.
Yeah, looks like they're quite common here, these benthic ptenophores.
That's some
Yeah.
nice hair. Not real hair. Wow, that's cool.
Yeah, great view of the, since you were speaking of the fishing line, though, those benthic ptenophores fishing.
Seems like there's maybe more of these like larger dead corals on this side than we were seeing on the other side. I'm not sure.
Perhaps.
Yeah, but this is quite common for the Madrapora. That's how they build reefs. They'll just topple over and then grow over themselves again.
Yeah.
Ooh, a viewer is asking, do you guys have any recommendations for websites that provide comprehensive information on various marine species identification?
Throw it over to Megan on that one. What do you use?
So I often use the OER Animal Guide, which is a bunch of pictures from the Okeanos Explorers Deep Discover ROV throughout their expeditions. The lab I work with at the University of Hawaii put together the imagery for that guide, and so we often use that as a reference when we're annotating these dives. There's also another website, the HURL Animal Guide, the Hawaii Undersea Research Laboratory Animal Guide, which is something that I coded for our website. It includes imagery from the Pisces 4 and the Pisces 5 submersibles, as well as the RCV 150 ROV and the Makali'i submersible that were operated by the University of Hawaii and the Hawaii Undersea Research Laboratory. Those subs were rated to 2,000 meters, so within this depth range, and so a lot of the animals that we're seeing here are actually in that animal guide. And I also put together a interactive web map for the Papahanaumokuakea Marine National Monument. If you Google PMNN animal guide or deep sea portal, you'll find that web page, and that will include imagery from Nautilus as well as from Okeanos Explorer and HURL, and all within the Papahanaumokuakea Marine National Monument. There's also an interactive web map that has the annotation data from all of the dives that were included in that study. So there's a lot of opportunity to find data, explore data in the deep sea. If you're also looking for deep sea data, you can check out repositories like the NOAA Deep Sea Coral and Sponge Data Portal. There's also OBIS, which is a repository of ocean data. So lots and lots of data out there, and we're continuously collecting more every day, like we're doing right now. When Harold, Jeff, and I get back to shore, we will be bringing a copy of the dives that we do on this expedition, and myself and the rest of us in the lab will be annotating the animals that we're seeing here, meaning we'll go through the video, rewatch it, and count and identify every single different type of coral, sponge, fish, and invertebrate, etc. that we've seen.
Thanks, Megan.
What's that?
Oh, what is that?
Oh, it's a siphonophore.
Go for a zoom
See, it's
there.
got the little swimming bells, and these are its feeding polyps. Boop, boop.
Oh. It's got a nice movement.
Yeah.
Is there something, some little associated on it?
It could be something it's eating.
Like on the left? Or
Yeah, I saw that.
It's a really nice view.
It's almost too close.
Yeah.
Now it is too close. I can't.
Yeah.
You can let it go.
Wait, it's back.
There it is. Maybe you could turn the lasers off to get some nice view of that without. Oh.
Dusting it.
Oh, another fun
Alright.
thing that viewers at home might be interested in, if you're really fond of these animals and want to learn them yourself and really like games, there is an app for your phone called Fathomverse. The Fathomverse game includes imagery from dives just like ours. It teaches you how to identify the different organisms, and then you become a little ROV and you fly around in this space and you find different animals, and if they're part of your mission, you collect them. And once you finish your mission, you get to sort those animals into the groups that you are targeting, and it will tell you if you've done a good job sorting them. And this will help train our models for AI as well. So by playing the game, you can contribute to deep sea science.
There's another fish.
Cutthroat eel.
Yeah, it's Iliophis, the arrow-toothed eels.
Nice basket star.
I love the basket stars. The way they move is just so fun.
Yeah, and good contextual information for the earlier collection, how many of these benthic ptenophores there are. They seem to be on all the Chrysogorgids.
Yeah, that's interesting, considering Steve was saying that was really uncommon. That might be something that's quite successful in this area.
I mean, I've seen them on Chrysogorgids before. It's just I haven't seen this many on every single Chrysogorgid we've seen before. That's impressive.
Yeah, but there's also the effect that once you see one and your eye gets the search image, and then you see them everywhere.
Oh, I had been seeing them quite a bit earlier. It's just we were doing other things.
What was the name of that app you mentioned, Megan?
It's Fathomverse.
Fathomverse.
Yeah, it's available for iPhone and Android.
And fun fact, video from Nautilus was also used in the development of that app. And not just Nautilus, but many other programs contributed data to the development.
You might actually find imagery from this dive on there in the future.
Is that a crinoid?
It is.
On the, yep.
This is Sathometra. It's a crinoid in the family Xenometridae.
Go for a zoom there.
They like to hold on to things. So those little legs are called cirri that are holding on to the bamboo coral.
Oh yeah, cool.
And it has a broken arm, but don't worry, it'll regrow it. This is an unbranched bamboo coral that we're looking at. You can see the polyps really nice and close here, and how it has these intertentacular spines, these little spines in between the tentacles. They're pretty short.
Nice.
Happy to zoom up.
That was cool.
A viewer is asking, do you ever find human litter or like plastic this deep in the ocean?
Absolutely, yes. We see trash a lot. and it's pretty sad.
Do we know if there's any presence of microplastics?
Yes, there is.
Wow.
So even in the deepest trenches, microplastics have been found in the guts of animals. Which is also very sad.
Yeah, I think that's the microplastics being everywhere and kind of invisible makes it kind of hard to see how much pollution there is, but when we do see objects on the seafloor, can be anything from a can to, we've seen even like whole massive recycling bins, which is kind of a strange occurrence, things that get washed away during storms or get thrown overboard, coolers, things like that have been seen even in the trenches, but yeah, microplastics is a big one. There's a lot of people studying that too.
I think earlier today in one of the shifts, they did find a rubbish at the bottom. I'm not sure if they collected it or not, but they did show it on camera.
I like this little pinnacle.
Yeah.
It's fun.
Okay, so a follow-up question on the plastics. What kind of impacts do macro and microplastics have on deep sea animals? And have we seen any animals using these plastics, like cans, as homes?
Yeah, on a previous expedition this year, one of the folks on the ship, Chris, regularly works on an atoll, Palmyra, and he shared a story about these large crabs that these coconut crabs that live on the island, that many of them will use anything for their shell, even like an old helmet or trash. And I think sometimes you'll have those moments where animals repurpose trash for something like that, but most of the time it's more of an issue with bioaccumulation. So animals filter feeding or eating, trying to consume what they're used to, and the water being filled with plastics, they end up accumulating all of those chemical toxins that are in microplastics, which can go up through the food chain.
Yeah, you know those little carrier crabs that we've been seeing everywhere? I've definitely seen some carrying trash before, which is a little sad.
What kind of things have you seen them carry?
Just like little pieces of line or like piece of metal. Some of the big ones, because there are larger species, and they'll pick stuff up.
So many colors of this rock.
But other things like nets can foul some of these corals. And we often see any substrate, any hard substrate especially, that's added to the deep sea environment gets colonized by animals. For example, shipwrecks or cars or other types of metal waste will often be covered by those red restingent sea stars. They like to get up off the seafloor, and it's a great little place for them to get up off the seafloor and do that. So the animals will take advantage of substrate. They're not super picky when it comes to what it actually is. They will colonize it and use it.
We got about 15 minutes left of bottom time.
Yep.
This is like really exciting with everything that we've seen so far.
Yeah, the bright colors definitely add a nice touch.
Yeah.
Thinking about it, where do these corals get their colors? Because corals, like in coral reefs, like they get their colors from their zooxanthellae. So where do they get their colors?
Yeah, so they have different pigments, so they do not have, obviously here there's no light, although that's a whole nother side story, but so yeah, they typically don't have zooxanthellae, these endosymbiotic algae in their tissues, although on some deeper waters that has been detected in lower numbers, but in terms of their coloration, yeah, they have different pigments in their tissues, and yeah, that begs the questions why why they have these certain colors, especially because for these organisms, and there is no light here, so why would something have a coloration? And a lot of speculation there. In some cases, it might be a byproduct of something that has a different function, but yeah, very interesting that you have these and then a common thing in some fishes and in shallower depths is that they are a bright red, and red, that's because that's a type of camouflage, is because red is the first light wavelength that is filtered out in the water, so having that is basically makes you dark. But yeah, at these depths there is no more light.
Yeah. Definitely an interesting topic to like look into.
Yeah, and it might be quite specific for each species. It's color at this depth for corals, and even at hydrothermal vents, you get these really bright red tube worm heads that, so, the color isn't necessarily for a purpose, but just sort of a byproduct of its biological features, and for each species it might be really different.
Yeah, thank you for that.
Yeah, and one more thing to add, yes, I talked about black corals, that's what I focused my work on, and there was one species we studied, and it had several dozen of different colorations, from bright white to bright pink, green, and we had to do the DNA to figure out that they were all exactly the same thing. But our eyes are so trained to the color is the first thing that catches your eye, and so yeah, to what Megan was alluding to earlier, that's it takes a long time to figure out what the characters are that are important for differentiating different species, genera, families, and so on, and that obviously requires observations and sample collections, and that's why these programs are so important. Collecting data. This is the very first time anyone has ever laid eyes on this seamount.
Oh, yeah.
It would be pretty unusual if everything we saw in the deep sea was just black and white. So I guess that's what people thought before coming down here with lights and cameras, though. At least some people did, but.
Yeah, and I think that brings up a really good point, is in the minds of most people, the deep sea is this uniform dark nothingness, emptiness, and
Yeah.
now when you look down from an airplane into the ocean, it all looks like this dark carpet of emptiness, and once you get to shine a spotlight on the bottom of the ocean, you see how much life there is in certain places and how important marine protected areas like the Palau National Marine Sanctuaries are, and as providing a safe haven for biodiversity, not just regionally but globally.
Some of the research I'm involved in off-ship relates to the creation of oceanic imaginaries and where those come from different cultural perspectives and even from sort of a media and film perspective, and how we've come to think of different parts of the ocean the ways that we do and how diverse that is across the world. And some of those really dominant narratives of vast, unknown, emptiness, scary, and the big one being the alien figure, are things I'm really interested in challenging, as we in this van are more the aliens than the inhabitants down here. And I think the emphasis on interconnectivity between deep ocean environments, shallow water, coastal water areas, and coastal communities and communities around the world should be more of the figure we focus on than the alien in these depths. But it's super interesting how these global imaginaries will create our perception of these spaces, and that perception will in turn really link our own relationship and how much we care about or are interested in protecting or conserving these spaces. So kind of looking at it from a more of a, maybe philosophy or cultural perspective about what these visuals really can do on a cultural side, I think really interests me.
Yeah, thanks for sharing, Ray.
There's a number of cultures around the world and ancestral relationships actually with deep ocean environments that did not require direct access to form understandings of these spaces as being vibrant and rich. So that I think that's pretty interesting, how diverse our ways of connecting with this space and how we perceive it as remote or interconnected.
Yeah, I think we'll running out of time here, of bottom time, so we'll be making our way up to the surface here in a bit, especially because we'll collect some samples on the way up. So coming off the bottom here at 1,000, and then we'll try to collect a Niskin bottle at 500, at 250, and 100. But yeah, I just want to take a second to thank everybody. It's been an extraordinary first dive of the expedition. Couldn't have asked for better terrain, better biodiversity. Thank all of our viewers online and our scientists ashore. and also acknowledging the generous support from our sponsors, the NOAA Office of Ocean Exploration and Research through the Ocean Exploration Cooperative Institute that provided funding for these expeditions to, for the first time, explore some of these deep water habitats within the Palau National Marine Sanctuary as a contribution that was by a commitment made by the U.S. government a few years ago to help map this vast area that exists in the Palau Exclusive Economic Zone. Yeah, great dive. We're also very pleased at how much terrain we covered, so looks like close to a kilometer and 1.3 kilometers maybe.
Yeah, we did complete our dive plan.
Nice Harmathiid. An anemone.
That's a Pheliactis. And there's another black coral. Lots of little anthomastus, little mushroom corals.
Looks like it's wishing us a happy Halloween.
Right? It's kind of reminding me of the plant from Little Shop of Horrors.
Yeah, exactly.
I love to peek underneath those ledges.
Oh yeah, peeking under ledges is great. There's always
Full of life.
little secrets under there.
Yeah. Oh.
Is it time? Seems
Just
like
about.
Yeah, I think so.
Yeah, I'll start getting set up. Will this be our recovery heading? Do you have any idea? Or
I think so. I'll ask the bridge how they feel.
okay. Start to come up a little bit off bottom.
Coming up.
I'm gonna take out this tether wrap here.
Where's the mouse? There it is.
Okay. You can turn off auto heading on Atalanta.
Oh, heading off.
Before we head up, are we able to take a one more Niskin for our 1,000-meter eDNA sample?
Sure.
Wait, no, we're not doing 1,000 meters because we triggered 2 Niskin at 1,050. So we're just going to do 500, 250, and 100.
Got it. Thanks, Mehani.
Okay. Scratch that. Come back up. Okay, we're gonna go tail to tail.
Bridge nav. We are off bottom and we should be on the surface in about 20 minutes. Yep.
Start coming up. Yep. Okay. Sorry. At I'm going about 22 meters a minute.
Okay.
Deck control.
Oh, I was waiting for the go ahead.
Sorry.
Jumped up.
Yeah, you can catch up, but get the deltas about even.
Okay.
0.
I'm gonna go a little faster.
Yeah.
Deck control.
Go ahead.
We are off bottom, heading up.
Copy, top bottom, heading up. Please let me
Roger.
So there's a question, when coming up from depths, do Herc and Atlanta have to go slow, like humans, to prevent bends? Are there reasons why you have to move a certain speed?
No, there's no limitation. We just, well, there is a limitation, but it has to do with the weight of the vehicle and how fast Hercules can thrust up, how much force it has, but not similar to a human where we don't have to worry about the pressure change.
And then there's another question from people watching. What are the deepest depths you all have worked at, and what were you researching? Back row? Oh, are there any of them left? Yeah.
Can you repeat that question?
Yeah, they were asking what are the deepest depths you all have worked at, and what were you researching?
For me, this is my first cruise on the Nautilus, so these are some of the deepest depths that I have worked at.
I think for me, was not on the Nautilus because this is my first time too, but it was on the polar stern, and it was up in the Arctic, and it was about 2,500 meters, and we were studying inverts larvae.
That's a much different climate.
It is very different and very cold.
How do you enjoy the cold?
I like it. I think it's pretty nice. I feel like it's a little warmer than inside the Nautilus because I am freezing in this control room. But it's pretty nice, yeah.
Yeah, we have to keep the control van cold to keep all the computer systems cold.
Yeah, I actually, before coming on the cruise, they told me that it was going to be really cold. So I ended up going through my Arctic clothes and packed a couple to bring here.
It's amazing.
Yeah.
What about you, Megan?
The deepest dive that I was navigating for was 5,300 meters in the clearing Clipperton zone. And that was with ROV Lu'ukai, the University of Hawaii's ROV, which is a 6,000 meter rated vehicle.
That's very cool. There's another comment. What a fantastic dive. The video stability is great, and the watch lead's knowledge has been top notch. Thanks, crew. Someone else asked, when nearing the end of the dive, has there ever been a time when you wanted just 10 more minutes? You just don't want to leave quite yet.
Oh yeah, we're always like, just a little bit longer.
Hey everybody, I'm just going to send an audio clip to shore really quick, so don't mind me saying strange things that are only relevant from hours ago.
When did we want to do our Niskins? What were the depths?
Can we do one at 500?
All right, yeah. We are going to be there soon.
Yes, thank you.
So this is the first dive of Lihu's voyage, and I'm so excited. Up to a couple hours ago, there was not even bathymetry of this site. So excited to see it in high resolution from the mapping, and then have the ROVs down here on the seafloor and really reveal, to humanize the very first time, the biodiversity of this place. It's been years in the making, and look at this amazing landscape. Okay, break, thanks.
Deck control, we are passing 500 meters. Yeah. Good morning.
Cold up here, huh?
It's really comfy in my onesie. We're at 5, yeah, we're recovering. Mike might need help on deck.
Okay, yeah, if you want to stick around here, I can go.
Yeah, I'll stick here and we'll finish this.
Okay.
For a Niskin.
250.
Okay.
Yes.
And then 50 for the last one?
No, it's 100.
100,
Yes.
okay. Right? I'm cozy. It even has pockets for my hands.
That's going to become your go-to van outfit,
I think so.
not just a Halloween thing.
Honestly, I think it's going to be my midnight to 4 outfit.
Don't think that's a bad idea. You might be too cozy, though.
Then I'll be ready for bed. I won't be cold and I'll already be dressed.
Ali, we have a question for the ROV pilots. Why is Atalanta deeper than Hercules? In a deeper position.
Atalanta is actually normally in a shallower position than Hercules.
Okay.
And we do that because Atalanta on top is like an eye in the sky and gives us some situational awareness with a video camera. And it's also attached to the ship in tension, so it's tight with the cable and decouples the ship from, the ROV Hercules from the ship motion, allowing us to get really smooth and picturesque video of the seafloor.
Awesome, thank you. And what's the distance between Hercules and Atalanta?
It depends, but normally anywhere from 15 to 20 meters. They're connected by a yellow tether that sometimes you'll see in the Atalanta image. And that tether is 30 meters long.
Thank you.
I'm gonna go to launch and recovery. So here, I think. Actually, I'll wait a little bit longer.
So just to update some of our new viewers that just came in, the ROVs are ascending, and can I ask the scientists to explain the different levels of, or the different areas where you collect the Niskin bottles?
So we collect water samples on the bottom at 2,500, 1,500, 250, and 100 meters. These numbers we came up with the numbers thinking that we would get different species along each depth. Which is why we picked them. Originally, we were supposed to do 10 meters, but Hercules needs time to be in recovery mode, and I think that starts at 50 meters, which is why we switched to 100. But yeah, that's the reason why we get the different depths, because we think we get different diversity for each step. Thank you.
Thank you.
That was a good dive. Yeah. Yeah, all right. We do have the winch on a big screen. Okay.
What is that? Oh my gosh.
Squid. Pan left. Okay, this will be the last one.
May, is that?
No, wait. Ooh. Squid. All right, Niskin number 6, fired, 100 meters.
Thanks, team.
Pan back right for me, if you can. And at 50 meters, we'll go out, stop on the winch. Hand it over the deck. What was it? Oh, actually, we should get it in our stowed position, which is straight ahead, racked all the way back, and looking down. Okay. Start turning off some of the lights. All right, I'll stop. 50 meters. So much squid.
Are they feeding?
Yeah, shut them down. Shut them down.
Deck control, all stop 50 meters.
Deck copies, all stop 50 meters. Break, bridge, back deck. Permission to go ahead and recover the ROVs.
Back deck, permission to recover, Charlie to recover.
Thank you, Captain.